Welcome to the deep dive. We're here to unpack complex ideas, really pull out the essential insights, and help you cut through all the noise to understand what truly matters. Today, we are diving headfirst into, well, honestly, one of the most fundamental and I think most hopeful frontiers in science right now, the biology of aging.
Yeah, absolutely.
And this isn't just about, getting older chronologically. It's about understanding the mechanisms that actually drive our decline and crucially, the groundbreaking medicine that's emerging to help us live not just longer, but, well, better.
It really is a field that's just utterly transformed in recent decades. I mean, it's moved from being quite abstract, almost theoretical, to rigorous testable science.
Right.
So our mission for you in this first deep dive really is to lay down a clear foundation. We need to define some critical terms, get everyone on the same page, and give you a real sense of how scientists are tackling this profound challenge of aging today.
And it is a challenge, isn't it? I mean, just picture this with me for a second. On one hand, maybe you have an 80-year-old guy. He's lean, sharp as a tack, and he doesn't just run. He completes this absolutely brutal 135-mile ultramarathon. In Death Valley, no less.
Wow.
Yeah, the oldest person ever to pull that off. Just an incredible picture of vibrant longevity.
Amazing.
But then, the stark contrast. Picture someone who's just 55, and they're already grappling with chronic diabetes, maybe debilitating heart disease, painful arthritis, basically experiencing major health burdens decades too soon.
Exactly. And these aren't just anecdotes, right? This variation is something we see everywhere. The data really underscore this disparity quite powerfully. We know that by age 55, a pretty significant chunk of adults, maybe 6 or 7%, are already battling 2 or more major chronic diseases.
2 or more by 55.
Yeah. And that figure, unsettlingly, it rockets up to nearly 1/3 of the population by the time they hit 70.
1/3. Goodness.
It means for a lot of people, the mid-50s aren't just a number. They can actually mark the beginning of a long, slow decline in health. And yet, as you said, we also see these octogenarians who seem almost youthfully vigorous. They completely defy the common expectations for their age.
So what is actually going on here? This huge variability, this massive difference in how people experience getting older, that's precisely what geroscience, the scientific study of aging, is trying to figure out.
Right.
The core question really becomes, why? Why do some individuals biologically age so much faster than others? And maybe even more importantly, how can we intervene? How can we shift everyone, not just the, genetically lucky ones, towards that healthier, more vibrant end of the aging spectrum?
And the stakes just couldn't be higher. I mean, globally, our population is aging at a rate we've never seen before.
Right.
Back in 2019, there were about 703 million people aged 65 or older worldwide. That number, it's projected to pretty much double to around 1.5 billion by 2050.
1.5 billion, that's hard to even grasp.
It really is. To give you a sense of that scale, by the middle of this century, roughly 1 in every 6 humans on the planet will be over 65. Compare that to about 1 in 11 back in 2019.
Wow.
And this massive demographic shift, it creates enormous challenges, especially when we look at something called the healthspan gap.
Healthspan gap. Explain that.
So on average, people around the world spend about the last 9.6 years of their lives in poor health. That's a period often riddled with chronic conditions, disability, just not living well.
Nearly a decade on average.
Yeah. And here in the United States, that gap is actually even wider. It averages around 12 years.
12 years.
12 years. So think about that. If you live to be 80 in America, statistically speaking, you can expect about 12 of those years to be significantly impacted by illness and disability.
That's sobering. So going back to those first examples, the 80-year-old ultramarathoner, he's basically managed to compress those years of infirmity down to almost nothing, perhaps.
Exactly. He's essentially squared the curve, as we say, compressed the morbidity. Meanwhile, that ailing 55-year-old,
he's already living in that period of decline right now.
Precisely. It's a really powerful, almost stark reminder of why we urgently need new solutions. And that really is the core mission driving this field and this deep dive, to understand and crucially to figure out how we might influence aging itself. This is just paramount if we want to close that health span gap.
It's not just about adding more years to life.
No, absolutely not. It's about ensuring people can live not just longer, but fundamentally better, healthier, more engaged lives. That distinction is absolutely central to everything we're going to talk about.
Okay. So with that vivid picture setting the stage, let's maybe set the compass for this exploration. It feels important to clarify right up front what this deep dive is, and maybe just as importantly, what it is not.
Good idea. Yeah.
First off, this is definitely not a collection of quick longevity tips for you to, start doing tomorrow morning.
No dual quick hacks here.
No. Nor are we going to give you a comprehensive tour of every single age-related disease like Alzheimer's, Parkinson's, specific cancers. Those are critical topics, absolutely, but they deserve their own dedicated deep dives, and we cover those elsewhere in this series.
And maybe most critically, we are absolutely not here to sell you anti-aging snake oil or promise immortality.
Yeah.
That's frankly the realm of fiction and a lot of unproven claims that we're going to steer completely clear of.
Here.
We also won't be treating aging itself as a formal disease in this particular deep dive. That reflects the current stance of regulatory bodies like the FDA. So you won't hear us claiming there's some magic pill for aging reversal out there.
None of that.
Our commitment is to ground this whole discussion firmly in the latest scientific evidence. And that means acknowledging the negative results, the field trials. Seeing those as valuable insights, not just, getting swept up in the hype.
Exactly. So what is this deep dive then? Well, think of this as your foundational big picture orientation to the actual science of aging. We're going to clarify what aging really means in precise biological terms. We'll explore how scientists are actually measuring aging, now going way beyond just counting birthdays.
Right.
We'll introduce you to the critical frameworks, things like the hallmarks of aging and the geroscience hypothesis that are really guiding all the cutting-edge research right now.
And we'll be looking at aging as a truly systemic thing. It's not just a collection of separate diseases happening randomly. It's a complex, interconnected process that affects your resilience across multiple organ systems, all at once, really.
Mm-hmm.
We'll give you a kind of roadmap showing how scientists design and conduct experiments to study aging, really focusing on the rigorous methods they use to try and prove cause and effect, not just, simple correlation.
Super important distinction.
Crucial. And we'll also provide an overview of how these fascinating discoveries are starting to be translated into potential therapies for actual human use.
So essentially, this episode is the groundwork. It's the sturdy foundation, right? Everything else in this series will build on this future deep dives, focusing on specific aging mechanisms, different types of interventions, and also the really profound ethical and societal implications that come with all this.
Yeah.
Our core objective for you, the listener, is that by the end of this, you should have a shared, grounded understanding of what slowing aging genuinely entails, and just as important, what it doesn't. You'll hopefully gain the principles you need to evaluate any supposed anti-aging therapy you hear about, helping you sort the real evidence from the wishful thinking.
Exactly. We want to equip you with the tools to stay grounded in that evidence, to critically assess things, even negative results and failed trials, because honestly, they often provide just as much, if not more, valuable insight as the big successes.
Okay, let's dive in then. The most basic question first. What is aging? It sounds simple, but biologically, it's actually quite nuanced, isn't it? It's more than just time passing.
Much more. Biologically, aging is commonly defined as the progressive loss of an organism's functional integrity over time.
Functional integrity?
Yeah, think of it as a gradual decline in the body's ability to maintain itself, to repair damage effectively, and to respond adequately to challenges. And this whole process is, inevitably accompanied by an increasing vulnerability to death.
Okay.
A more succinct way to view it is as the accumulation of cellular and molecular damage. Our bodies are constantly under assault, facing wear and tear, and while we have these really remarkable repair mechanisms, over time, the damage simply accumulates faster than our systems can fully counteract it. It just builds up.
The balance shift.
Exactly. And this leads to that gradual loss of function across various systems and, critically, an increased risk of developing a whole host of diseases. What's really fascinating, though, is that it's not a uniform process. It varies hugely between individuals and even within one person. Well, it often starts remarkably early in life, much earlier than people think. And different cells, different tissues can age at quite different speeds. It's all influenced by this complex mix of your genetics, your environment, lifestyle choices, and honestly, sometimes just random chance.
Right. So how can we make that more tangible? Is there a practical way to think about this decline?
Yeah, a really useful way to conceptualize it, to make it sort of concrete for you, is to think of aging as a decline in what scientists call resilience or homeostatic reserve.
Homeostatic reserve,
Yeah.
like a buffer.
Exactly like a buffer.
Yeah.
When we're young, our bodies have this incredible buffer zone. They can easily absorb stress, bounce back from damage, maintain a stable internal environment that's homeostasis. You get sick, you recover quickly, you injure yourself, you heal up fast.
Right. Kids bounce back from everything.
They really do. But as we age, that buffer starts to thin out. That robust capacity to recover from stressors gets significantly reduced. An older person might seem perfectly fine just sitting there at baseline, but then you introduce a challenge. Maybe it's a surgery, common infection like the flu, or even just an environmental stress like a heat wave. And that challenge, which a younger person might barely notice, can lead to a major, sometimes lasting setback for the older individual.
Because their reserve is lower.
Precisely. Their system just doesn't have the same capacity to buffer that stress and return to normal.
Is there an example you often use to illustrate this?
Yeah, a powerful one used often in gerontology involves comparing two hypothetical 80-year-old women. Let's say they both undergo the same standard knee replacement surgery.
Okay.
One of them sails through recovery. She's back doing brisk walks within weeks, fully regains her independence, basically back to her old self.
Great outcome.
Fantastic. But the other woman, same age, same surgery, she suffers postoperative delirium, maybe develops complications like an infection, and maybe never quite regains her full independence or her baseline functional capacity. She's permanently impacted.
Wow. Same age, same procedure, vastly different outcomes.
Exactly. And the crucial point here is that their chronological age alone, both being 80, didn't dictate those vastly different paths.
So what did?
It was primarily their biological age, maybe combined with a bit of luck, that played the crucial role. Biological age, in this context, describes the accumulated wear and tear on their cells, the erosion of that resilient margin of safety that allows the body to maintain function under strain.
Got it.
So really, at its core, aging can be fundamentally understood as the loss of systemic resilience over time.
That makes a lot of sense. Okay, now, you mentioned healthspan earlier. Let's distinguish that clearly from lifespan. They sound similar, but they're critically different, right?
Absolutely vital distinction. Lifespan is simple. It's just how long you live, your chronological duration on Earth. End of story.
Okay.
Healthspan, on the other hand, is the period of your life that you spend in good health, largely free from significant chronic disease or disability. It's the years you're functional, vibrant, engaged.
The quality years, essentially.
The quality years, exactly. Now, over the past century, we've seen incredible advancements. Public health, sanitation, nutrition, medical treatments. And these have done a truly remarkable job of dramatically extending human lifespans. We've added decades to average life expectancy globally.
Huge success story in many
Not
areas.
deniably. However, extending health span has proven to be a much, much more elusive goal. In many respects, what we've often done is simply trade those acute early killers, like infectious diseases that killed people young,
Right?
for a prolonged period of chronic late-life illnesses, like heart disease, diabetes, dementia, arthritis.
So we live longer, but maybe not necessarily better for those extra years.
That's the crux of the issue. On average, as I mentioned, people worldwide spend about the last decade of their lives roughly 9.6 years in poor health. And in some populations, like the U.S. with its 12-year gap, that unhealthy period is even longer and potentially growing.
So the main goal of, let's call it successful longevity science, isn't just pushing lifespans to 120 or 50.
Not primarily, no. The core aim is really to achieve what's known as compressing morbidity.
Compressing morbidity, meaning squeezing the bad part.
Exactly. The idea is to delay, or ideally even prevent, the onset of those chronic illnesses and disabilities that are associated with aging, so people stay healthy and functional for a much longer proportion of their lives. And the period of decline, if and when it happens, is significantly shortened.
So the ideal scenario, the squaring the curve idea you mentioned.
Right. The ideal is something like you live to, say, 100, maybe 110, with minimal chronic illness, maintaining your physical and mental vigor for almost all of it. And then you experience a very brief, relatively rapid end-of-life decline. You compress that period of sickness and disability into the shortest possible window right at the very end.
Instead of it dragging on for 10 or 15 years. Okay, let's do a quick micro recap here about 5 minutes in. We've established that aging, biologically, is this complex systemic loss of resilience over time. It's distinct from just counting years. And the key challenge, the focus of geroscience, is extending health span, those healthy, functional years, not just total lifespan.
Perfectly summarized. That distinction between lifespan and health span and the goal of compressing morbidity is absolutely foundational.
Great. So that brings us perfectly to the next major concept we need to unpack, the geroscience hypothesis. You mentioned geroscience. What's the core idea behind this hypothesis? It sounds like a pretty big deal.
It is a big deal. It's really the guiding framework behind most modern efforts to extend health span, and it truly represents a paradigm shift in how we approach medicine.
Okay.
At its core, the geroscience hypothesis proposes that aging itself, the underlying biological processes, that sort of ticking time bomb, if you like, is the common root cause of most of the chronic diseases that plague us in later life.
Wow. Okay. So instead of seeing heart disease and Alzheimer's and cancer as totally separate things.
Exactly. Instead of viewing them in these isolated silos, each needing its own specific treatment, geroscience suggests they are, in many ways, different manifestations of one overarching process. The fundamental breakdown of resilience and repair that happens as we age.
That's a radical shift.
It is, and it has profound implications. What it suggests is that by targeting those fundamental shared mechanisms of aging,
The root causes.
yes, the root causes, we could potentially prevent or delay many different diseases all at once. We could achieve broad spectrum benefits for health span.
So instead of playing whack-a-mole with individual diseases.
Right. Instead of whack-a-mole, you're aiming at the underlying mechanism that makes the moles pop up in the first place. As one key paper put it, since aging physiology plays a major role in many, if not all, chronic diseases, therapeutically addressing aging physiology directly will prevent or mitigate multiple chronic diseases simultaneously. It's a move away from medicine's traditional one disease at a time focus.
That makes intuitive sense. Where did this idea first really gain traction? Was it from human studies?
Interestingly, no. The concept really emerged quite strongly from studying much shorter-lived lab organisms, animals like mice, worms, fruit flies.
The usual suspects in biology labs?
Exactly. In these model organisms, scientists observed again and again that specific interventions, whether genetic tweaks or environmental changes like diet, could not only extend their total lifespan, but also concurrently delay or significantly reduce the occurrence of multiple different age-related pathologies.
Can you give an example?
Sure. Take, for instance, a genetic intervention in mice that dampens down a key growth pathway called the IGF-1 pathway. These mice don't just live longer on average, they stay healthier across the board for longer. They develop less cancer, they show reduced cardiac fibrosis, their immune function stays better for longer. It's a holistic improvement, not just dodging one specific bullet.
So it's not just lifespan, it's health span extension too.
Precisely. That's geroscience in action in the lab. Not just avoiding one particular disease, but achieving a more general postponement of decline across many different systems. Another great example is the drug rapamycin in mice. It extends life, but it also seems to postpone almost all causes of death, pushing back the entire spectrum of age-related diseases.
Okay, that's compelling in mice. But the huge question, obviously, is, does this translate to humans? Are we seeing this play out in people?
That is the critical question, and this is where things get really exciting, because large-scale human tests designed specifically around the geroscience hypothesis are only just beginning to give us potential answers.
Like what?
Well, a landmark initiative, currently in the advanced planning stages, is the TAME trial. TAME stands for Targeting Aging with Metformin.
Metformin. That's a common diabetes drug, right?
Exactly. It's widely used, well understood, generally safe. The TAME trial is designed explicitly as a proof of concept for the geroscience hypothesis in humans.
How does it work?
It plans to enroll thousands of older adults who do not have diabetes. They'll be randomly assigned to take either metformin or a placebo.
Okay.
And here's the crucial part, the primary endpoint. What they're measuring isn't just the effect on one specific disease. Instead, they'll measure the time until a participant experiences any event from a predefined composite of major age-related diseases.
Not composite. What's included?
Things like a major cardiovascular event, heart attack or stroke, the diagnosis of invasive cancer, the onset of dementia, or death from any cause, whichever of those happens first.
So they're looking for a delay in any of those major age-related outcomes.
Precisely. This is a really bold and significant trial design, partly because the FDA had to agree to this novel endpoint.
Why is that significant?
Because, as we said, aging itself isn't currently recognized by the FDA as a disease you can get a drug approved for. So, for them to greenlight a trial aiming to delay a composite of age-related diseases using a drug targeting fundamental aging mechanisms, that's a strong signal. It shows the medical establishment is cautiously but definitely starting to embrace the geroscience idea.
So the trial design itself reflects the hypothesis.
Absolutely. The TANE trial's composite outcome design directly mirrors the geroscience hypothesis's need to capture global, multi-system effects rather than being limited to just single disease outcomes. It's designed to test if intervening and aging itself can push back the whole cluster of late-life diseases.
Okay, another quick micro recap, maybe around the 10-minute mark. We've just covered the geroscience hypothesis. The idea that treating aging itself, the root cause, could prevent multiple chronic diseases at once. This is strongly backed by animal models, and now it's being put to the test in humans with pioneering studies like the TANE trial using metformin.
Exactly. It's a fundamental shift, moving from disease-specific treatments to targeting the underlying aging process that drives them all.
So if we want to target aging, we need to know what exactly is going wrong. This brings us to the hallmarks of aging. You mentioned this framework earlier. It sounds like a sort of biological parts list for decline.
That's a great way to put it, yeah. The Hallmarks framework really tries to answer the question, what fundamentally goes wrong in our cells and tissues as we get older?
Where did this framework come from?
It was really crystallized in a landmark scientific paper published back in 2013 by Dr. Carlos Lopez-Oting and colleagues. They proposed that aging isn't just some random collection of failures, but is driven by a set of conserved underlying biological mechanisms.
Conserved meaning they appear across different species.
Generally, yes. Mechanisms that seem to be fundamental to the aging process in many organisms, including us. That original paper identified 9 hallmarks, and the framework just took off, becoming incredibly influential in guiding research.
And you said it was updated recently.
Yes, exactly a decade later, in 2023. The original authors, along with other key researchers, published an update, expanding the list to 12 canonical hallmarks. This revision really reflects the significant scientific advances and the deeper understanding we've gained over that decade.
12 hallmarks now. Can you give us a sense of what they are? Maybe run through the list.
Absolutely. Think of these as the key processes. So first, we have genomic instability. This is basically the accumulation of damage to our DNA mutations, rearrangements, that kind of thing.
Okay. DNA damage. Makes sense.
Second, telomere attrition. These are the protective caps on the ends of our chromosomes. They tend to shorten each time a cell divides, eventually limiting how many times cells can replicate.
Right, the cell division limit.
Third is epigenetic alterations. These are changes not to the DNA sequence itself, but to how genes are switched on or off. Think of it like changes to the software controlling the DNA hardware. Things like DNA methylation patterns change with age.
The control system gets scrambled.
In a way, yes. Fourth is loss of proteostasis. Proteostasis means protein maintenance. As we age, our cells become less efficient at making sure proteins are folded correctly and getting rid of damaged ones. This leads to buildup of misfolded or aggregated proteins, which you see in diseases like Alzheimer's.
Protein quality control fails. Got it.
Fifth, disabled macroautophagy. Autophagy is the cell's recycling system. It breaks down and clears out old, damaged components. As we age, this vital cleanup process becomes less effective, leading to accumulation of cellular garbage.
So waste builds up inside cells?
Exactly. Sixth is deregulated nutrient sensing. This involves distortions in key metabolic pathways that sense nutrient availability, like the insulin IGF-1 pathway or mTOR, AMPK, sirtuins. These are pathways we know can be tuned by diet or drugs to actually affect lifespan in model organisms.
The body's fuel gauge gets wonky.
That's a good analogy. Seventh is mitochondrial dysfunction. The mitochondria are the powerhouses of our cells. With age, they tend to falter, producing less energy and more damaging byproducts like reactive oxygen species.
Energy production declines.
Eighth is cellular senescence. We touched on this earlier. These are cells that stop dividing but don't die. They become zombie cells. They hang around and secrete a nasty mix of pro-inflammatory and tissue-damaging factors.
The zombie cells causing trouble.
Ninth, stem cell exhaustion. Our bodies rely on stem cells to regenerate and repair tissues. With age, these stem cell pools can become depleted, or the stem cells become less functional, impairing our ability to heal and maintain tissues.
Repair crews get tired.
Pretty much. 10th is altered intercellular communication. The way cells talk to each other changes with age. Often this involves an increase in pro-inflammatory signals circulating throughout the body.
Communication breakdown.
11th, closely related, is chronic inflammation. This used to be considered part of altered communication, but now it's recognized as a distinct hallmark. It's this low-grade, persistent, sterile inflammation, often called inflammaging, that accompanies aging and contributes to many diseases.
Just constant low-level inflammation.
All right. And finally, 12th is dysbiosis. This refers to age-related shifts in the composition and function of our gut microbiome, the trillions of microbes living in our digestive tract. These changes can impact metabolism, immunity, and overall health.
Even our gut bugs age with us.
They certainly seem to. So that's the current list of 12.
That's a comprehensive list. It covers genetics, epigenetics, protein handling, energy waste disposal, cell fate repair, communication, inflammation, even the microbiome. It really highlights how complex aging is.
It absolutely does. And what's crucial to grasp is that these hallmarks aren't operating in isolation. They are deeply, deeply interconnected. They form this complex web of interactions.
Can you give an example of that interconnection?
Sure. For instance, that genomic instability, the DNA damage we mentioned first, that can directly trigger a cell to become senescent, hallmark 8. Then those senescent cells start secreting inflammatory signals, contributing to hallmarks 10 and 11. Those signals can disrupt the function of nearby stem cells, contributing to hallmark 9. So you see this cascade effect. One thing goes wrong, and it pulls others down with it. Aging is truly multi-systemic because these processes influence each other.
It's like a web where pulling one strand makes the whole thing wobble.
That's a perfect analogy.
Now, you mentioned that for something to be considered a hallmark, it has to meet strict criteria. It's not just something that happens during aging.
Exactly. This is critical for understanding why these are considered potential targets for intervention. The criteria are quite rigorous. First, yes, the process must manifest during normal aging. It has to be something that typically happens as we get older.
Okay, that makes sense.
Second, if you experimentally aggravate that process, make it worse in a lab model, it should accelerate aging or aging-related diseases.
So making it worse speeds up aging.
Right. But the third criterion is arguably the most important, especially from a therapeutic perspective. Experimental amelioration of that process, making it better, should retard the normal aging process. It should slow aging down or improve health span.
So fixing it has to actually slow down aging. That's the key for intervention potential.
That is the absolute key. It implies causality and points towards a potential therapeutic strategy.
Can you illustrate that third point with an example?
Let's take telomere attrition. We know telomeres shorten with age. If you genetically engineer mice to lose their telomeres much faster, they show signs of premature aging. So aggravating it accelerates aging, criterion 2.
Okay.
Now, for criterion 3, there have been experiments where scientists use gene therapy to restore telomerase, the enzyme that lengthens telomeres, in already aged mice. What they found was an improvement in tissue function, better health span, and even a modest extension of lifespan. So ameliorating telomere shortening retarded aspects of aging.
So telomeres fit all 3 criteria.
They do. Another example is autophagy, the cellular recycling system. We know it declines with age. Experimentally boosting autophagy in various animal models generally tends to improve their longevity and resilience. So again, ameliorating it slows aging. This third criterion is what separates a mere correlate of aging from a potential driver and therapeutic target.
Got it. And are these 12 hallmarks set in stone, or is the list still evolving?
It's definitely evolving. Science is always refining its understanding. In fact, the 2023 update that expanded the list from 9 to 12 also discussed several emerging hallmarks processes that are strong candidates, but maybe don't meet all the criteria with the same level of evidence yet.
Whoa, like what?
2 notable ones they discussed are altered mechanical properties of tissues and splicing dysregulation.
Tissue stiffness and splicing.
Yeah. Altered mechanical properties refers to things like increased tissue stiffness or loss of elasticity in the extracellular matrix, the scaffolding between our cells. Think about lung fibrosis, hardening of the arteries, arteriosclerosis, even wrinkled skin. These mechanical changes happen with age and seem to influence how cells behave.
So the physical environment of the cells changes.
Exactly. And then splicing dysregulation refers to age-related drift in how our genes are spliced together to make messenger RNAs, which then make proteins. If the splicing goes wrong, you can get aberrant proteins or lose the diversity of proteins your cells can make. This has been observed in aged muscle and brain tissue and is thought to contribute to functional decline.
Interesting. So the list might grow further.
It very well might. The field is dynamic. The fact that the framework was updated after a decade shows that aging research is a moving frontier, constantly refining our understanding based on new evidence. It's not static.
Okay, let's pause for another micro recap around the 15-minute mark. We've just covered the hallmarks of aging, now numbering 12 key biological processes, ranging from genomic instability and telomere shortening to chronic inflammation and dysbiosis. These are thought to actively drive the aging process. they're interconnected, and critically, they meet rigorous causality criteria, making them potential targets for interventions aimed at slowing aging. We also noted the list is still evolving.
Excellent summary. Understanding these hallmarks gives us that parts list, the specific mechanisms we might try to influence to improve health span.
Which brings us to a really practical challenge, measuring aging. If aging is this complex, multidimensional thing with all these hallmarks interacting and varying between people, how on earth do scientists actually quantify it? How do you measure someone's biological age or track if an intervention is working?
That's a fantastic question, and it's an area that has seen a true measurement revolution in the past decade or so. Historically, as you can imagine, measuring aging in humans was pretty crude.
How did they used to do it?
Well, they relied on pretty general proxies. Things like a frailty index, which combines measures of weakness, slowness, weight loss, etc. Or they might look at levels of single inflammatory markers in the blood, like C-reactive protein or IL-6, or simply track the prevalence of age-related diseases in a population. Useful, but not very precise and personalized.
Kind of indirect measures.
Very indirect. But over the last 10-15 years, we've seen this explosion in the development of multi-parameter biomarkers of aging, and most famously, the creation of what are often called aging clocks.
Aging clocks. Like a clock that tells you your biological age instead of the time.
In essence, yes. The most celebrated and well-studied of these are the epigenetic clocks. These use those patterns of DNA methylation, the chemical tags on DNA we mentioned as an epigenetic hallmark, to predict age.
How do they work?
Basically, researchers analyze DNA methylation levels at hundreds of thousands or even millions of sites across the genome in thousands of people of different known ages. Then, they use machine learning algorithms to identify a specific subset of these methylation sites whose patterns collectively change very predictably with chronological age.
So they found the DNA locations that reliably change methylation levels as we get older.
Exactly. The original groundbreaking clock was developed by Steve Horvath back in 2013. His mathematical formula used the methylation levels at just 353 specific genomic sites. And remarkably, it could predict a person's chronological age with incredible accuracy. The correlation was around 0.96, typically with an error of only a few years. It worked across many different tissues and cell types.
Wow, that's incredibly accurate just from DNA tags. But if it just predicts chronological age, what's the big deal? We already know how old someone is.
That's the key point. The real scientific interest wasn't just its ability to match your calendar age. It was what happened when the clock's prediction deviated from your actual age.
When the biological clock was faster or slower than the calendar.
Precisely. This phenomenon became known as epigenetic age acceleration, if the clock said you were older than you are, or deceleration, if it said you were younger. Researchers quickly started investigating. Does this deviation actually mean anything for health?
And does it?
Overwhelmingly, yes. Study after study showed that having a higher epigenetic age than your chronological age, being epigenetically older, consistently correlates with a higher risk of developing numerous age-related diseases, like cardiovascular disease, cancer, cognitive decline, and critically, a significantly higher risk of all-cause mortality.
So if your epigenetic clock is running fast, it's a bad sign for your future health.
It certainly seems to be a strong risk indicator. For example, being 10 years older, according to some of these clocks, was linked to a substantially elevated risk of dying sooner.
Okay, so the original clocks predicted chronological age, and deviations predicted risk. Did the clocks get more sophisticated?
They did. The next generation of clocks moved beyond just predicting chronological age. Researchers started training them on different outcomes, specifically health outcomes and mortality.
How did they do that?
Well, one key example is DNAM-Finode, developed by Morgan Levine and her colleagues. Instead of training the clock on chronological age, they trained it using methylation data regressed against a composite phenotypic age score. This score was derived from various clinical blood chemistry biomarkers plus mortality data. So PhenoAge was designed to capture physiological dysregulation and predict health outcomes better than the first-gen clocks.
So it's trained on health markers, not just age.
Right. And then came arguably the most powerful predictor developed so far, DNAM Grimmage.
Grimmage. Sounds ominous.
It does, doesn't it? It was introduced in 2019 by Oclero, Steve Horvath, and others. Grimmage was constructed in a clever two-step process. First, they used methylation data to predict the levels of certain blood proteins known to be associated with risk, and also predicted smoking history from methylation patterns. Then they combined these predicted surrogate markers into a final score designed to predict time to death. So Grimmage was explicitly built as a mortality predictor.
Wow. Directly trained to predict lifespan. How well does it work?
It's remarkably predictive. In one large meta-analysis, even after controlling for chronological age and other standard risk factors, each standard deviation increase in a person's Grimmage score corresponded to about a 1.8-fold higher hazard of dying from any cause.
1.8 times the risk. That's huge. That's stronger than many traditional risk factors, isn't it?
It is indeed. Grimmage also correlates strongly with the development of various diseases and with the total number of chronic conditions a person has, their comorbidity count. The name is fitting. If your Grimmage calculation comes out, say, 5 years older than your actual chronological age, that's generally considered pretty grim news regarding your long-term health prospects.
Okay, so epigenetic clocks like Grimmage are powerful predictors. Are there other types of biological clocks besides epigenetic ones?
Oh yes, definitely. Epigenetic clocks got a lot of the initial attention, but researchers are developing biological age metrics based on all sorts of data types. There are proteomic clocks, which analyze the levels of dozens or hundreds of proteins in your blood. There are transcriptomic clocks based on gene expression patterns, RNA levels. There are metabolomic profiles looking at small molecule metabolites.
So clocks based on proteins, RNA, metabolites.
Right. And people are also building composite indices that mix some of these molecular markers with more traditional clinical or functional measures. You also hear about simpler, more organ-specific measures like calculating brain age from MRI scans or immune age based on the profile of different immune cell types in the blood. It's a very active area of research trying to find the best, most informative ways to measure biological age.
Fascinating. Now, you mentioned earlier that not everyone ages at the same speed. Is there a way to measure that pace rather than just the accumulated age?
Yes, and that's another really innovative and important concept, measuring the rate or pace of aging. It's clear some people accumulate biological damage faster than others.
Right.
The most prominent example of a tool designed to measure this is the Dundon pace algorithm. This came out of the fantastic Dundon longitudinal study in New Zealand, which has followed over 1,000 people since their birth in the early 1970s.
How does Dundon pace work?
It also uses DNA methylation data, but instead of predicting an age in years, it derives a value that represents the rate of biological aging, essentially. How many years of biological wear and tear a person is accumulating per one chronological year.
So it gives you a speed, like miles per hour, but for aging.
That's a great way to think about it. The average pace is calibrated to 1.0. So if your Dundon pace score is 1.0, you're aging biologically at the average rate. If your score is, say, 1.1, the algorithm suggests you're aging about 10% faster than average. If it's 0.8, you're aging more slowly.
And why is measuring the pace so useful?
What's really exciting about a pace measure like Dundon pace is that it seems to be more sensitive to short-term changes. This makes it potentially very valuable for intervention studies where you want to see if a treatment is actually slowing the aging process now rather than waiting decades to see if people live longer.
Ah, so you could potentially see if an intervention is working much faster.
That's the hope. And in fact, the first ever randomized controlled trial, RCT, to show a statistically significant change in a validated aging biomarker in response to an intervention used donepezil. This was the calorie trial, published in 2022.
Calorie. What did they test?
They tested the effects of moderate caloric restriction, CR, about 25% fewer calories than usual, in healthy, non-obese adults over a 2-year period.
And what happened to their pace of aging?
The group practicing caloric restriction showed a statistically significant slowing of their Dundon pace clock compared to the control group who ate normally. The effect size suggested roughly a 2-3% slower pace of biological aging on average in the CR group.
Wow. So CR actually slowed down their measured rate of aging.
It did, according to this specific biomarker. Interestingly, standard epigenetic clocks like Grimmage and Finage didn't show significant differences between the groups in this relatively short 2-year time frame. This suggests Dundon pace might indeed be a more sensitive early readout surrogate for interventions aiming to slow aging.
What are the implications of that finding?
Well, first, it's a landmark proof of concept. It provides the first RCT evidence in humans that the rate of biological aging, as measured by a validated biomarker, can be altered by an intervention. It gives strong support to the geroscience hypothesis in humans. Now, admittedly, 2-3% slowing is a modest effect, maybe translating to 1 or 2 extra healthy years if sustained over a lifetime. But demonstrating any measurable slowing in humans is hugely important scientifically.
It shows it's possible.
Exactly. It opens the door. It suggests Dundon pace could be a valuable tool for testing other potential geroprotective therapies much more quickly.
Okay, let's do another quick micro recap here around the 20-minute mark. We've covered the revolution in measuring biological age, moving beyond simple proxies. Epigenetic clocks, especially advanced ones like Grimmage trained on mortality, provide powerful predictions of health risks. And crucially, new measures like Bundin pace allow us to track the rate or pace of aging, offering a way to validate interventions like caloric restriction much earlier in human trials.
Precisely. This ability to measure aging accurately and dynamically is absolutely critical for moving the field forward towards real therapies.
Which leads us right into the next practical hurdle: translating these measurements and potential therapies into actual approved medicine. This involves navigating the complex regulatory world, particularly the distinction between surrogate endpoints and clinical endpoints.
Yes, this is a hugely important topic for anyone interested in how anti-aging therapies might actually reach patients.
So let's start with definitions. What exactly is a clinical endpoint in a trial?
A clinical endpoint is a direct measure of how a patient feels, functions, or survives. It's something intrinsically meaningful to the patient's health and well-being. Examples would be the actual incidence of heart attacks, strokes, or cancer diagnosis, measures of physical independence like ability to walk or care for oneself, or the most definitive endpoint, mortality, whether people live longer.
Okay, the stuff that really matters to patients. So what's a surrogate endpoint?
A surrogate endpoint is different. It's a biomarker, or some intermediate outcome that's used in clinical trials as a substitute for that direct clinical endpoint.
A substitute? Why use a substitute?
The key reason is efficiency. Measuring a true clinical endpoint, especially for chronic diseases or aging, can take many years and require thousands of participants. Think about proving a drug prevents heart attacks. You need to follow many people for a long time.
Right.
A surrogate endpoint, like a biomarker that changes much faster, allows trials to be shorter, smaller, and therefore much less expensive and time-consuming. The assumption underlying a surrogate is that changes in that marker reliably predict changes in the meaningful clinical outcome we actually care about.
Can you give a classic example of a successful surrogate?
Sure. LDL cholesterol, the bad cholesterol, is a widely accepted surrogate endpoint for cardiovascular disease. We have strong evidence from many trials that drugs lowering LDL cholesterol also reduce the risk of heart attacks and strokes. So, new cholesterol drugs can often get approved based on their ability to lower LDL, even before massive long-term outcome trials are fully completed. Blood pressure is another similar example.
Okay, so surrogates can speed things up dramatically. What's the downside? What's the danger?
The big danger is that the surrogate might not truly capture the clinical benefit. A therapy could successfully improve the surrogate marker, lower the number on the lab test without actually making patients live longer or better. Or, even worse, it could improve the marker while causing unforeseen harm through other mechanisms.
The marker improves, but the patient doesn't, or even gets worse.
Exactly. There are cautionary tales in medicine. A famous one involves HbA1c in diabetes. HbA1c measures average blood sugar over months, and it's a good predictor of diabetes complications. So it was assumed that aggressively lowering HbA1c by any means necessary would automatically lead to better long-term outcomes.
Makes sense.
But some large clinical trials, like the Accord trial, found that aggressively forcing HbA1c down to very low levels in high-risk patients using multiple drugs actually increased cardiovascular deaths compared to less aggressive control.
Whoa. Lowering marker led to worse outcomes.
In that specific context, yes. It was a wake-up call. It showed that simply improving the surrogate, HbA1c, didn't guarantee improvement in the clinical endpoint survival, likely due to off-target effects or the risks of the aggressive polypharmacy itself. It proved that not every predictive biomarker makes a valid surrogate endpoint for drug approval. You have to be really careful.
So that brings us back to the aging clocks and biomarkers we just discussed. Where do they stand in this regulatory landscape? Are any of them accepted surrogate endpoints?
As of right now, no. No epigenetic clock, no proteomic clock, no other complex aging biomarker has been formally validated and accepted by regulatory agencies, like the FDA, as a surrogate endpoint for aging or age-related diseases.
None of them, despite how predictive Grimmage is.
Despite how predictive they are observationally, yes. The bar for surrogate validation is incredibly high, and rightfully so.
What does that validation require?
Regulators, like the FDA, generally insist on robust evidence demonstrating that a treatment-induced change in the biomarker reliably predicts a treatment-induced change in the hard clinical outcome. It's not enough to just show correlation in observational studies. You need data from intervention trials showing that when the drug moves the marker, it also moves the clinical outcome in the same beneficial direction.
And we don't have that yet for aging clocks.
We simply don't have those large, long-term human intervention trials yet that definitively show, for example, that making someone's epigenetic age younger with a specific therapy translates into measurably fewer diseases, better function, or longer life because of that therapy. We lack that causal link demonstrated in humans through intervention.
So what's the FDA's stance on targeting aging then? Are they just saying no?
Not exactly no. They're actually quite open, conceptually. Their position, based on workshops and discussions, seems to be: we're willing to consider aging as a potential target for interventions if the field can rigorously define how to measure it, and crucially, if you can prove that modifying that measure leads to tangible clinical benefit for patients.
So the door is open, but the burden of proof is high.
Very high. In the meantime, while we wait for potential surrogates to be validated, the advice from both the FDA and the National Institute on Aging, NIA, has been for researchers to pursue other strategies in clinical trials, like using multi-outcome composite endpoints, as we discussed with the TAME trial, or focusing on function-based endpoints, things like walking speed, grip strength, cognitive function tests, measures that directly reflect how well an older person is functioning.
So measure things that directly matter to quality of life.
Exactly. The TAME trial's composite endpoint is a perfect example of trying to capture that broad geroscience-based benefit without relying on an unvalidated surrogate. They're aiming for a claim like delays the onset of multiple major age-related morbidities.
Are there downsides to using composite endpoints like that?
There can be challenges. Sometimes combining multiple different types of outcomes can dilute the effect on any single one, making it harder to show statistical significance. It can also complicate regulatory review because the different components of the endpoint might fall under the expertise of different divisions within the FDA, e.g., cancer drugs versus heart drugs.
So it's not a simple solution either.
No, it requires careful design and early conversations with regulators to agree on the trial structure and what would constitute success. It might even require creating entirely new regulatory pathways or indications that reflect this multi-disease preventative approach.
Okay, so there's a difference between a biomarker being useful for research and being accepted as a surrogate for drug approval. Is there an intermediate step?
Yes, there is. The FDA has a biomarker qualification program. Through this program, a biomarker can be formally qualified for a specific context of use in drug development.
Context of use?
Yeah. For example, a biomarker might be qualified as a pharmacodynamic marker, showing the drug is hitting its biological target, or as a prognostic marker, predicting disease course, or for patient stratification, identifying who might benefit most. Some aging-related biomarkers, like measures of cellular senescence, for example, P69NK4A, or inflammation, for example, IL-6, are being explored through this qualification process.
But qualification isn't the same as surrogate approval.
Critically, no. Qualification means the FDA agrees the biomarker is reliable for its specified purpose within a trial, but it doesn't mean that changing that biomarker is sufficient grounds for approving the drug. Surrogate endpoint status, allowing approval based on the biomarker change alone, is a much, much higher bar that requires that strong link to clinical outcomes.
So how might an aging biomarker eventually become a surrogate?
The likely pathway is that large outcome trials, like TAME, will first have to succeed in showing clinical benefit based on hard endpoints, like the composite disease outcome. Then, researchers can go back and retrospectively analyze which biomarkers changed significantly in tandem with that clinical benefit in the treatment group. Consistent findings across multiple successful trials could then build the case for qualifying that biomarker as a future surrogate. But it's a long road.
Okay, so bottom line on regulation. Regulators are interested, they see the potential, but they rightly demand rigorous proof of actual clinical benefit in humans before approving drugs to treat aging. Biomarkers are exciting tools, but none are shortcuts to approval yet. Composite endpoints and functional measures are the current strategies for trials like TAME.
That's a perfect summary. The science of aging biology is definitely becoming mainstream, but it still has to earn its place in the clinic through solid, patient-centered evidence.
Now, related to proving things work, let's talk about proving causation in aging research. We've touched on this, but it's so fundamental. How do scientists move beyond just observing correlations like older people have more senescent cells to proving that a specific factor actually causes aging or age-related decline?
This is absolutely central. Because if you want to develop an effective therapy, you need to be targeting a cause, not just a symptom or an unrelated correlate. We mentioned the criteria for a hallmark, requiring that fixing it should slow aging that inherently points towards causality. But let's dig into the specific experimental strategies used to really nail down that causal link.
Okay, what are the gold standards?
There are several classic experimental designs used across biology to test if X causes Y, and they're heavily used in aging research.
Right.
One very powerful strategy is ablation, which basically means removal or elimination. The logic is simple. If factor X is causing aging Y, then getting rid of X should slow down or prevent Y.
Remove the cause, remove the effect.
Precisely. The experiments clearing senescent cells in mice are a prime example of ablation. Researchers created genetically engineered mice where senescent cells uniquely express a suicide gene.
A suicide gene?
Yeah, a gene that makes the cells susceptible to being killed by a specific, otherwise harmless drug.
Yeah.
So by giving these mice the drug, they could selectively eliminate the senescent cells.
Clever. And what happened?
The results were quite striking. In mice that were genetically engineered to age rapidly, progeroid mice, periodically clearing their senescent cells delayed the onset of multiple age-related problems like cataracts, muscle wasting, osteoporosis, and significantly extended their median lifespan.
Wow. So removing senescent cells helped in fast-aging mice. What about normal mice?
The similar positive effects, though maybe less dramatic. In normally aging mice, lifelong periodic clearance of senescent cells led to them being healthier and more active in old age, less cancer, better kidney function, more resilient, and also resulted in a modest but statistically significant extension of their median lifespan.
So the ablation experiments strongly suggested senescent cells aren't just bystanders, they actively contribute to aging dysfunction.
Exactly. They provided strong causal evidence that senescent cells are drivers, not just correlates of aging.
So ablation is one strategy. What else?
Another powerful approach is the rescue experiment. This often involves taking an organism that is aging abnormally fast due to a specific known defect, and then intervening to try and fix or bypass that defect. If the intervention normalizes the aging process or extends lifespan, it provides strong evidence that the targeted mechanism is causally involved.
So you take a broken system and see if fixing a specific part helps.
Right. For example, there are mouse models with mutations in DNA repair genes like ERCC1. These mice suffer from accelerated aging. They go gray, lose weight, become frail, and die very young, mimicking aspects of human progeroid syndromes.
Okay.
If you then treat these fast-aging mice with an intervention, maybe it's a stem cell therapy, or a xenolytic drug to clear the damaged cells, or maybe something that boosts NAD plus metabolism to support repair, and that intervention significantly rescues their health, improves function, and extends their short lifespan. That's strong evidence. It implies the pathway you targeted is indeed causally linked to the aging process, both in this accelerated model and likely in normal aging too.
Got it. So ablation removes a factor, rescue fixes a defect. What else?
Showing a dose-response relationship is also important. If increasing the dose of a potential geroprotective intervention leads to a greater benefit up to an optimal point, of course, before toxicity kicks in, it supports a causal effect.
More drug, more effect, up to a point.
Exactly. The studies with rapamycin in mice often show this. Generally, higher doses within a tolerable range, and starting the treatment earlier in life lead to larger extensions in lifespan compared to lower doses or later starts. This consistency supports the idea that rapamycin is working through a specific causal mechanism related to mTOR signaling, which is dose-dependent. Of course, you also see trade-offs. Very high doses in mice can cause side effects like diabetes symptoms.
Right. The dose makes the poison or the cure.
Always.
Yeah.
And finally, a really crucial type of evidence in aging research is cross-modal evidence, or what's sometimes called triangulation.
Triangulation, like finding a location from multiple points.
Precisely that idea. If you have several entirely different types of interventions, all thought to converge on the same underlying biological pathway or hallmark, and they all produce similar benefits, like extending lifespan or health span, it dramatically strengthens the case for causality. It makes it much less likely that the effect is just an artifact of one specific drug or genetic manipulation.
Give me an example of triangulation.
Let's go back to that nutrient sensing pathway, specifically the IGF-1-MTO signaling axis. We know that multiple very different interventions reduce signaling through this pathway. You have caloric restriction, dietary change. You have genetic knockouts of growth hormone or IGF-1 receptors, genetic change. And you have drugs like rapamycin, a pharmacological intervention.
3 totally different approaches.
Right. And remarkably, all 3 of these independent approaches consistently extend lifespan and improve health span in mice. Conversely, interventions that boost this pathway, like chronic overfeeding or certain gene overexpressions, tend to shorten lifespan. This convergence of evidence from multiple angles gives scientists very high confidence that nutrient sensing, particularly via this IGF-1/mTOR pathway, is a genuine causal mechanism involved in modulating the aging process, not just some fluke observation tied to one specific method.
That makes a lot of sense. It builds a much stronger case. Can we apply this logic to another hallmark, say mitochondrial dysfunction?
Absolutely. We know mitochondrial DNA mutations accumulate with age. So first, aggravation. Scientists engineered mice that accumulate these mtDNA mutations much faster, the so-called mutator mice. And sure enough, these mice show accelerated aging phenotypes like premature graying, osteoporosis, frailty, reduced lifespan. That indicates mitochondrial integrity is crucial.
Okay, aggravation speeds aging.
Then amelioration. Interventions aimed at supporting mitochondrial function, things like regular exercise, potentially NAD, plus precursors like NR or NMN, though human data is still evolving there. Maybe certain gene therapies targeting mitochondrial pathways often show benefits for healthspan or function in animal models, and sometimes hints in human studies. So you have evidence from both making it worse and trying to make it better, pointing towards mitochondria being causally involved in aging.
So it's about building this weight of evidence from different angles.
Exactly. Especially in aging research, where definitive, decades-long human intervention trials are so difficult and expensive, we often rely heavily on this weight of evidence approach. Consistent findings across different species, using different experimental models, genetic, dietary, pharmacological, and demonstrating effects like ablation, rescue, dose response, and triangulation. That's what collectively shifts a hypothesis from mere observation towards accepted causation.
Okay, so we've established how scientists build a strong case for causation, often in animal models. But then comes the big hurdle. From mice to men and women, the translation trap. You've alluded to this already. Just because something wonders in a mouse doesn't mean it will work in us. Why is this translation so difficult?
This is the perennial challenge, the source of much frustration and, honestly, many failed clinical trials in medicine, not just in aging research. Despite meticulous causal proof in animals, translating discoveries to humans is just fraught with pitfalls. There are many reasons for these translation failures.
What are some of the main ones?
Well, first and foremost are the fundamental species differences. Mice are just not tiny humans. Their basic physiology is radically different.
How different?
Think about it. A mouse heart beats around 600 times per minute. Ours is maybe 60, 80. Their body temperature is higher, around 39 degrees Celsius. And their natural lifespan is only 2-3 years compared to our 80 or so. These are huge differences.
Yeah, fundamentally different operating systems.
Right. And while many core aging pathways are conserved between mice and humans, their relative importance, or the specific ways they manifest and interact, can differ significantly.
Like the telomere example you gave earlier.
Exactly. Mice have active telomerase in most of their body cells throughout life, whereas adult humans mostly don't. This means telomere shortening is likely a much more critical driver of cellular aging in humans than it is in mice. So a telomerase activating therapy might show little benefit in a normal mouse, but could potentially be very beneficial for humans. Conversely, something like a general antioxidant might help a short-lived mouse combat oxidative stress, but do absolutely nothing in humans because we have evolved much more robust, long-lasting antioxidant defense systems.
Okay, species differences are huge. What else trips up translation?
Genetic uniformity versus diversity. Lab mice used in experiments are often highly inbred. They're genetically almost identical, like clones. This reduces variability in experiments, which is good for statistical power.
But humans aren't like that.
Not at all. Human populations are incredibly genetically diverse. This means a drug or intervention might work great in one specific inbred mouse strain, but its effects in humans could vary wildly depending on an individual's unique genetic background. There can be complex interactions between the intervention and different genetic profiles. This is one reason the NIA's Interventions Testing Program, ITP, which tests potential anti-aging drugs, now deliberately uses genetically heterogeneous mice to better mimic human diversity.
That makes sense. What about differences even within mice?
Yes, even within the same species, you see significant strain and sex differences. An intervention might work brilliantly in one strain of mice, but do nothing in another. And as we touched on earlier, sex differences can be profound.
Like the acarbose example.
Precisely. Acarbose extended male mouse lifespan by 22% in one ITP cohort, but only 5% in females, which wasn't always statistically significant. Another striking example is 17-alpha-estradiol, a form of estrogen. It increased median lifespan by 12-19% in male mice across multiple studies, but did absolutely nothing, zilch, for female lifespan.
Why would that be?
The thinking is that females likely already have optimized pathways related to estrogen signaling due to their own endogenous estrogen, so adding more doesn't help or might even be detrimental. The implication for humans is clear. Some interventions might need to be tailored or at least tested specifically for males versus females. We can't assume what works for one sex will work for the other.
Okay, species, genetics, sex. What about the practicalities, like dosage?
Dose metabolism. This is a huge one. Mice metabolize most drugs much, much faster than humans do, often requiring relatively higher doses per kilogram of body weight to achieve a therapeutic effect. If you simply scale that mouse dose up to a human based on body weight, the required amount might be impractically enormous or dangerously toxic.
Like the resveratrol example, maybe.
That was part of the issue, yes. But rapamycin is another perfect case study. The doses needed for robust lifespan extension in mice cause relatively mild side effects in mice. But human trials using rapamycin, or its analogs, rapalogs, for anti-aging purposes typically use much lower, often intermittent, e.g. once-weekly doses. Why? To minimize the known risks in humans, like immune suppression or metabolic disruption, like inducing diabetic symptoms. The big unanswered question then becomes, can humans safely take a high enough dose for long enough to achieve the same kind of dramatic benefits seen in mice? We don't know yet.
So finding the right dose window in humans is critical and difficult.
Absolutely. And then there's the husbandry and environment. Lab animals live in highly controlled artificial environments. They're usually kept in super clean cages, often pathogen-free, fed a standardized chow diet protected from predators and harsh weather. It's a cushy life compared to ours.
Right, humans live out in the wild, relatively speaking.
Exactly. We face infectious challenges, variable diets, psychological stresses, environmental toxins. An intervention that works wonders in a sterile lab bubble might fail or even be harmful in the real world.
Can you give an example?
Caloric restriction, CR, is a good one.
Yeah.
In the controlled lab environment, CR dramatically extends mouse lifespan. But imagine putting a wild mouse on severe CR. It might become weaker, less able to forage or escape predators, and more susceptible to infections. The net effect in a natural environment could easily be negative.
That makes sense.
Even in the primate CR studies, the difference between the Wisconsin and NIA results was partly attributed to the control diets. The NIA control monkeys were already on a healthier portion-controlled diet than the Wisconsin controls who ate more freely. So while CR was still beneficial for health span in both studies, its effect on lifespan extension seemed less dramatic in the NIA monkeys, possibly because their baseline diet was already pretty good. This highlights the importance of context and the need for replication. It also warns us against assuming an intervention will add huge benefits if people are already practicing healthy lifestyles.
So the controlled lab environment might exaggerate benefits sometimes.
It certainly can, or mask potential downsides that only appear under real-world conditions. And finally, there's the simple fact of short lives versus long lives. A mouse lives maybe 3 years. A 10% lifespan extension is only about 3-4 months, relatively easy to detect in a reasonably sized experiment. Humans live around 80 years. A 10% extension is 8 years, proving that definitively would require trials lasting decades. It's just not practical for initial testing. This is why there's so much focus on shorter-term health span outcomes, or those surrogate biomarkers we discussed, hoping they predict long-term benefits.
Okay, so species differences, genetic diversity, sex differences, dose issues, artificial environments, and the sheer difference in lifespan scale all contribute to this translation gap.
Exactly. It's a minefield.
And you mentioned resveratrol as a cautionary tale earlier. Can we unpack that a bit more? It got so much hype.
It really did. Around 2006, studies showed resveratrol could extend the lifespan of obese mice fed a high-fat diet, and it activated sirtuins, which were thought to be key longevity enzymes. This created a massive stir. Headlines proclaimed a fountain of youth compound found in red wine.
I remember that. Everyone was talking about red wine.
Yeah. But the reality turned out to be much more nuanced and frankly disappointing. Firstly, resveratrol did not significantly extend the lifespan of normal, healthy mice on a standard diet. The benefit seemed restricted to counteracting the negative effects of obesity.
Yeah, okay, that's a big difference.
Huge. Secondly, subsequent human trials, and there have been many, have been largely underwhelming. While some studies showed minor metabolic improvements, like better insulin sensitivity, at very high doses, resveratrol supplements haven't delivered the transformative anti-aging effects initially hoped for. Part of the issue was likely the dosage. The equivalent human dose based on the mouse studies would require drinking hundreds of glasses of red wine daily. But part of it might just be that human biology responds differently, or that targeting sirtuins alone isn't the magic bullet people thought it was.
So the lesson learned is?
The lesson is, don't jump to conclusions based on single animal studies, especially those under specific conditions like obesity, or based purely on mechanistic insights without robust outcome data. Maintain a critical eye, demand replication, and wait for human evidence.
Which reinforces the need to learn from failures and limitations.
Absolutely. We need to actively seek out and discuss the null results, the contradictory findings, the failed trials. That's how science self-corrects.
Like the Unity senolytic trial you mentioned.
Exactly. Unity Biotechnology had very promising preclinical data showing their senolytic drug cleared senescent cells and improved osteoarthritis in mice. Mechanistically, it made perfect sense. But their phase 2 human trial in patients with knee osteoarthritis, reported in 2020, unfortunately failed to show any significant improvement in pain or function compared to placebo over 12 weeks.
That must have been a blow.
It was, and it certainly reined in some of the initial, maybe overly optimistic exuberance about senolytics being an imminent cure for arthritis. The company shifted focus. It doesn't mean senolytics are dead. Maybe the drug wasn't potent enough, or the trial duration too short, or osteoarthritis is just a very tough nut to crack, but it was a crucial reality check.
Any other examples of high-profile disappointments?
Well, the whole field of young blood, or parabiosis, has faced challenges translating from mice to humans. Parabiosis, surgically joining the circulatory systems of old and young mice, showed dramatic rejuvenation effects in the old mice. This led to intense interest, and even clinics offering infusions of plasma from young human donors to older individuals.
Right, the vampire therapy idea.
They're sort of.
Yeah.
But controlled clinical trials, for instance, looking at infusions of young plasma in Alzheimer's patients, have largely failed to show significant improvements in cognition or function. The effects, if any, were far less dramatic than the mouse parabiosis results suggested. Again, it highlights that human aging might be much harder to manipulate, or perhaps requires much more targeted, precise interventions than just flooding the system with youthful factors. These failures are just as important as the successes for guiding future research.
Okay, so given this complex landscape of basic science, translation challenges, and mixed results, let's try to map out the current intervention landscape. What are the main categories of approaches being actively pursued right now?
We can broadly group the current interventions into maybe 4 main buckets, ranging from the well-established to the highly experimental.
Okay, bucket number 1?
Bucket number 1 is lifestyle and behavior. This is the oldest, most proven, and arguably still the most impactful category for most people right now.
What's in this bucket?
The 2 heavy hitters are caloric restriction, CR, and exercise. CR, as we've discussed, is the gold standard in virtually every species tested in the lab. It robustly extends both lifespan and healthspan.
But hard for humans to stick to.
Extremely hard long-term. Severe CR can have side effects like loss of muscle mass, reduced bone density, feeling cold, loss of libido. It's not practical or appealing for most. However, even shorter-term studies of moderate CR in humans, like the calorie trial, show clear benefits. Improved cardiometabolic risk factors, a younger metabolic profile, and, as we saw, even a slowing of an epigenetic aging clock by 2–3%. Whether it extends maximum human lifespan is still unknown, but mechanistically, CR seems to positively influence many of the hallmarks: nutrient sensing, mitochondrial efficiency, autophagy, inflammation.
So CR is powerful but difficult. What about exercise?
Exercise is arguably the closest thing we currently have to a true, widely accessible anti-aging intervention. It might not dramatically extend maximum lifespan, but it absolutely, unequivocally extends health span.
How so?
People who are physically active and maintain good fitness throughout life have significantly lower rates of cardiovascular disease, type 2 diabetes, many cancers, dementia, disability, and frailty in older age. Exercise acts like a systemic tonic. It keeps your muscles strong, bones dense, heart healthy, metabolism efficient, brain sharp, and even seems to help maintain genomic stability and reduce inflammation. It touches almost all the hallmarks in a beneficial way.
And it works even if you start later in life.
Yes. That's another key benefit. Exercise is one of the few interventions shown to extend even median lifespan in mice when started relatively late in life. It's never too late to benefit.
So diet, like CR principles, may be less extreme, and exercise are the foundation.
Absolutely. They are the foundation for any sensible longevity regimen, and they set the benchmark that any new drug or therapy really needs to beat, or at least add significantly to. We know adherence to strict CR is an issue. So pharmacological approaches might aim to mimic some of CR's benefits or target pathways that lifestyle doesn't hit as effectively. But the data linking healthy behaviors, not smoking, healthy diet, regular exercise, moderate alcohol, healthy weight to significantly longer life expectancy, potentially adding up to a decade or more, is undeniable.
Okay, so lifestyle's bucket 1. What's bucket 2?
Bucket 2 is pharmacological interventions, basically. Drugs or supplements aimed at targeting aging pathways. This is where a huge amount of current research and investment is focused.
We've mentioned a few already, like rapamycin and metformin.
Exactly. Rapamycin and its analogs, rapalogs, target that central MTR pathway involved in growth and metabolism. They have repeatedly and robustly extended lifespan in mice, making them probably the most validated lifespan-extending drug in mammals so far.
In human trials.
Human trials are cautiously underway, mostly using lower intermittent doses. A key early trial by Joan Manick and colleagues in 2014 showed that an mTOR inhibitor, everolimus, given for just 6 weeks, significantly enhanced the immune response to a flu vaccine in older adults by about 20%. It also reduced the proportion of aged dysfunctional T cells.
Interesting. Boosting immune function.
Yeah. Another company, Restore Bio, ran larger trials with a similar drug, RTB101. Their phase 2b trial showed promising results, significantly reducing the incidence of respiratory tract infections in high-risk elderly individuals by 30–50%. However, their subsequent large phase 3 trial unfortunately failed to meet its primary endpoint in 2019.
Why did it fail? Does that kill the idea?
Not necessarily. The failure could have been due to various factors. Maybe the trial design, maybe an unusually mild flu season during the trial, maybe issues with a specific drug formulation or dose. It certainly tempered expectations, but the underlying biology targeting mTOR for immune rejuvenation remains compelling. There are ongoing trials looking at low-dose rapamycin in pet dogs, the Dog Aging Project, a fascinating large-scale study, and smaller human trials exploring its effects on things like immune function, skin aging, and even early Alzheimer's disease.
Okay, so rapamycin is a major contender. What about metformin?
Metformin, as we discussed regarding the TAME trial, is another top candidate. It's cheap, widely used for diabetes, has a great long-term safety profile. The intriguing epidemiological findings diabetics on metformin sometimes living longer than matched non-diabetics really fueled interest in its potential general health benefits beyond just blood sugar control.
How might it work for aging?
The mechanisms aren't fully clear, but it seems to impact several hallmarks. It can reduce insulin levels, influence nutrient signaling pathways like AMPK, potentially reduce inflammation, and maybe even have direct effects on mitochondrial function. Small trials have shown benefits in some age-related conditions like reducing frailty markers or inflammation.
But you mentioned a potential downside with exercise.
Yes, the MASTERS trial finding that metformin seemed to blunt muscle mass and strength gains from resistance training in older adults is an important consideration. It appeared to dampen the normal mTORC1 signaling response in muscle needed for growth. It's a reminder that these drugs aren't necessarily a free lunch. There could be trade-offs, and the context, like combining with exercise, matters. Physiology is complex.
Fair enough. What other drugs are in this bucket?
There are quite a few others generating interest. Acarbose, another diabetes drug that slows carbohydrate digestion, also extended mouse lifespan significantly, especially in males in the ITP studies. NAD plus precursors, like nicotinamide riboside, NR, or nicotinamide mononucleotide, NMN, aim to boost levels of NAD plus apin, a crucial molecule for energy metabolism and DNA repair that declines with age. The goal is to support mitochondrial function and potentially enhance repair processes. Human data is still accumulating and somewhat mixed here.
Right, the NAD plus boosters.
Then you have senolytics, the drugs designed to selectively eliminate those senescent zombie cells. Dasatinib, a cancer drug combined with quercetin, a plant flavonoid, is one commonly studied combination. Others are in development. As we saw with the Unity trial, translation to clear human benefit is still a work in progress, but the concept remains very appealing.
Clearing out the bad cells.
Exactly. Antioxidants have a more checkered history. The classical broad-spectrum antioxidants like high-dose vitamin E, vitamin C, beta-carotene largely failed in big prevention trials, and some even showed increased risk of certain cancers. The focus now is shifting towards more targeted approaches, like compounds that activate the NRF2 pathway, which is the body's own master regulator of antioxidant defenses. For example, sulforaphan from broccoli, curcumin from turmeric.
So activating internal defenses rather than just flooding the system.
That seems more promising. Anti-inflammatory agents are also being explored, like low-dose drugs targeting specific inflammatory cytokines, for example, IL-1 or IL-6, to combat inflammaging. But again, potential trade-offs with immune suppression are a concern. Even common drugs like aspirin and statins, widely used for cardiovascular disease prevention, might have broader geroprotective effects, though the jury is still out on widespread primary prevention use in healthy older adults due to risks like bleeding with aspirin.
What about hormones? There was a lot of hype around HGH in the past.
Yeah, hormonal modulators have a controversial history. Things like DHEA or human growth hormone, NGH, were touted back in the 90s, often without solid evidence and with significant potential risks. More recently, the small TRIUMPH trial generated headlines by claiming that a combination of growth hormone, DHEA, and metformin reversed epigenetic age by about 2 years in a small group of middle-aged men over 1 year. Intriguing, but it was a tiny, uncontrolled pilot study, and GH has known side effects. Needs much larger controlled validation.
Lots of possibilities in the drug bucket. Are there others?
Oh, yes. People are looking at specific peptide therapies that might mimic youthful factors or induce regeneration. Microbiome modulators, probiotics, prebiotics, even fecal transplants are being investigated to restore a more youthful gut environment. It's a very active area. The NIA's intervention testing program alone has identified around 13 different compounds, mostly repurposed drugs or natural products that reliably extend mouse lifespan, typically by 5–15%. That provides a strong pipeline of candidates for further investigation.
Okay, lifestyle pharma. What's bucket 3?
Bucket 3 is where things get really advanced. Advanced biological therapies, primarily focusing on cells and genes.
This sounds more futuristic.
It is, but research is moving fast. One area is stem cell replacement or revitalization. The idea is to replenish or rejuvenate the body's declining stem cell pools to improve tissue repair and immune function. This might involve infusions of hematopoietic stem cells for the blood system or mesenchymal stem cells, which can go to various tissues. Results in humans have been mixed so far, often hampered by issues with cell delivery and survival. A related concept is taking a person's own cells, rejuvenating them in the lab, perhaps using techniques we'll discuss, and then giving them back.
Using your own younger cells.
Potentially. Then there's gene therapy. This involves delivering specific genes into the body to counteract aging processes. We mentioned the mouse study, where a single gene therapy delivering telomerase, HDR, extended median lifespan by about 24% and improved health, even when given to adult mice. That was a landmark finding suggesting telomere rejuvenation could be beneficial systemically. There have also been anecdotal N1 self-experiments by some researchers using viral vectors to deliver genes like telomerase or follistatin, which promotes muscle growth. Highly controversial and risky, but pushing boundaries.
Delivering longevity genes directly.
That's the idea. But perhaps the most paradigm-shifting concept in this bucket is partial cellular reprogramming.
The reprogramming we touched on earlier, this sounds like the most sci-fi.
It really does. So full cellular reprogramming using the famous Yamanaka factors can turn any adult cell back into an age 0 embryonic-like pluripotent stem cell. That's amazing for research, but you can't do it directly in a living person because it would cause tumors, teratomas. The breakthrough idea with partial reprogramming is to only transiently or cyclically express some of these reprogramming factors, like 3 of them, OCTI4, SOX2, KLF4, often called OSK. The goal is to rejuvenate the cells, reset some of the epigenetic aging marks without completely erasing the cell's identity and making it pluripotent.
Just turn back the clock a bit, not all the way to 0.
Exactly. That landmark 2020 study from David Sinclair's group showed this could work functionally. They used an OSK gene therapy delivered by a virus to regenerate damaged optic nerve cells in old mice and even restored vision in a mouse model of glaucoma. That was a direct reversal of age-related functional loss in the nervous system.
Restoring vision in old mice. That's incredible.
It really is.
Yeah.
And more recent work from 2022 and 2023 has shown that applying cycles of partial reprogramming systemically in mice can reverse molecular markers of aging in multiple tissues and improve regenerative capacity in muscle and kidney. It's still very early days, definitely bleeding edge stuff, and there are risks doing it wrong can cause cancer or even kill the animals, but the potential is staggering if this can be safely controlled.
It could literally reset epigenetic age in our cells.
That's the ultimate vision. Perhaps periodic gene therapy infusions that act like a system-wide epigenetic reset button, pushing back aging at the cellular level. It's still speculative, but it's arguably the most potentially transformative approach being explored.
Wow. Okay, so lifestyle, pharma, cells, and genes. Is there a bucket 4?
We could maybe add a 4th bucket for other novel devices or technologies. It's a bit more diverse.
Like what?
It could include things like organ-specific regenerative approaches. Maybe technologies aimed at regrowing or rejuvenating the thymus gland to restore youthful T-cell production using tissue engineering or hormone therapies. Or maybe plasma exchange devices technologies being explored to filter old plasma to remove potentially harmful pro-aging factors that accumulate. Companies like Ambrosia briefly offer this, and research groups are still investigating plasma filtration or dilution more rigorously.
Cleaning the blood, essentially.
In a sophisticated way, yes. It could also include continuous monitoring and intervention devices. Imagine implantable devices that could stimulate nerves, like the vagal nerve to reduce inflammation, or deliver drugs on a precise schedule, or advanced biofeedback devices that help people optimize their physiology based on real-time data. It's a broader category for tech-based solutions.
Okay, that gives us a great overview of the intervention landscape. It's vast.
It is. And just to reiterate, as we go through this series, we'll be connecting these different interventions back to the specific hallmarks of aging they target. We'll always try to critically evaluate the evidence, especially the evidence in humans, distinguishing biomarker changes from real functional improvements that meet a minimally clinically important difference, MCID, and we'll constantly highlight the risks, the unknowns, and the fact that most of these are not ready for prime time. This is not a recommendation for self-experimentation. The goal is rigorously tested, approved, safe therapies for everyone.
Absolutely. Okay, so with all this research and potential therapies, what is the current regulatory status? You said aging isn't officially a disease, so how would a drug targeting aging actually get approved?
That's the billion-dollar question, really. And the current reality is, as we've stated, no drug currently carries an indication from the FDA or EMA, European Medicines Agency, to prevent aging or extend lifespan. Aging per se is not listed as a disease they approve treatments for.
So it's still seen as a natural process, not a medical condition?
Broadly speaking, yes, by the regulators. This creates that fundamental challenge. How do you get a drug approved if its target isn't a recognized disease with established clinical endpoints?
But you mentioned subtle shifts are happening.
Yes, there are definitely movements. For instance, the World Health Organization, WHO, in its latest International Classification of Diseases, ICD-11, introduced a new code, XT9T, for aging-related conditions. This can be used to tag conditions where aging itself is considered the main causal factor, particularly pathologically accelerated aging. It's a small step, but symbolically important.
Okay, WHO is acknowledging it more directly. What about the FDA?
The FDA stance, as we discussed, seems to be one of cautious openness. They've indicated they are willing to consider approval and a potential indication if a clinical trial convincingly demonstrates that an intervention can increase healthy lifespan or delay a broad set of age-related impairments. They're not closing the door, but they need the data proving broad clinical benefit.
So how might the first approvals actually happen in practice? Would it be an anti-aging label?
Probably not initially. The first approvals are more likely to come through a sort of back door.
How so?
For example, if the TANE trial is successful, if it shows metformin significantly reduces the composite endpoint of major diseases, metformin might receive a new indication. Wouldn't say prevents aging, but maybe something like indicated for the prevention of major age-related morbidities in non-diabetic older adults, or wording to that effect.
So focused on preventing the diseases of aging.
Exactly. Or another scenario. Imagine a senolytic drug gets approved for a specific disease where senescent cells are clearly implicated, like idiopathic pulmonary fibrosis, lung-scarring disease. Once it's approved for that narrow indication, doctors could potentially prescribe it off-label for broader age-related conditions where senescence is suspected to play a role, although that comes with its own issues regarding evidence and reimbursement.
So initial approvals might be disease-specific, with broader use potentially following off-label.
That's a likely path for some therapies. The regulatory conversation is also happening in Europe. The EMA has explicitly asked the question, should EMA consider aging a disease? There are arguments being made in favor, but currently their focus is more on ensuring medicines are adequately tested in geriatric populations, rather than targeting aging itself as an indication.
Is there any intermediate condition that might serve as a stepping stone?
Frailty is often discussed as a potential stepping stone. Frailty is a recognized geriatric syndrome characterized by weakness, weight loss, exhaustion, slowness. It's clearly age-related and predicts poor outcomes. There have been clinical trials targeting frailty as an endpoint, though none have led to an approved drug specifically for frailty yet. But success in a frailty trial could potentially pave the way towards broader aging-related indications.
So frailty might be an easier target than aging overall.
it might be seen as more clinically defined and measurable in the short term. And we also have the ongoing efforts, like the FD-NIH BEST program, working on formally qualifying aging biomarkers like epigenetic clocks or proteomic scores for specific uses in trials, as we discussed. But again, qualification doesn't equal surrogate approval.
So it really is uncharted territory from a regulatory perspective.
It truly is. If and when the first definitive trial of a therapy targeting fundamental aging mechanisms shows clear, broad, positive results in humans, regulators will essentially have to figure out how to label it. It might be a complex indication listing multiple prevented diseases, or perhaps eventually a whole new category recognizing geroprotection.
And in the meantime, companies often focus on single diseases.
That's often the pragmatic strategy right now. Target a specific age-related disease where the mechanism is clear, like fibrosis or a specific neurodegenerative disease, get approval for that, and then hope the broader anti-aging potential becomes apparent through further research or real-world evidence, potentially leading to expanded use.
So the bottom line on regulation is the science has to lead the way. Solid evidence of clinical benefit is needed before regulators can formally recognize and approve treatments targeting aging itself.
Absolutely. The ball is firmly in the court of the scientists and clinical trialists to provide that rigorous evidence.
And this huge effort isn't just about individual health or scientific curiosity. There's a powerful argument that successfully slowing aging would have massive benefits for society as a whole. This brings us to the concept of the longevity dividend. What's that about?
The longevity dividend is a term popularized by researchers like S. Geroscience, Dana Goldman, and others. The core concept is incredibly compelling. Even a small slowdown in the fundamental biological process of aging would yield absolutely astronomical financial savings and quality of life improvements across the entire population.
Astronomical savings? How does that work?
It stems from the fact that age-related chronic diseases, heart disease, cancer, diabetes, Alzheimer's, osteoporosis, etc., account for the overwhelming majority of healthcare expenditures and lost productivity in developed nations. They're the main drivers of suffering and cost in later life.
Right. Most healthcare spending happens in the later years dealing with these diseases.
Exactly. Now, imagine an intervention that doesn't just treat one of these diseases, but targets the underlying aging process and manages to delay the onset or slow the progression of multiple major age-related diseases simultaneously, even by just a couple of years.
The geroscience approach.
Precisely. The effects would compound dramatically. Instead of just preventing deaths from heart disease only to have people die from cancer or Alzheimer's a few years later, the competing risks problem,
right,
you'd be pushing back the onset of all of them, leading to a longer period of healthy, productive life for everyone.
Has anyone tried to quantify this dividend?
Yes. Dana Goldman and colleagues published an influential study in Health Affairs back in 2013, doing just that. They modeled a hypothetical intervention that slowed aging enough to increase average life expectancy by just 2.2 years. Critically, most of those added years were assumed to be healthy years due to the delay in disease onset.
Okay, a modest 2.2-year healthy life extension. What was the estimated economic value?
Their estimation was that this modest improvement would be worth approximately $7.1 trillion in economic value to the United States alone over a 50-year period.
$7 trillion from just over 2 extra healthy years.
Trillion with a T.
Yeah.
That figure includes savings on healthcare costs, the value of extended workforce participation, and also attempts to monetize the intangible value people place on living longer in good health.
And how did that compare to, say, curing cancer?
That was the striking part. Their model showed that even completely curing cancer or heart disease would yield far smaller economic gains than this modest slowing of aging. Why? Because of those competing risks. If you cure cancer, people live longer but then are more likely to get and require treatment for heart disease or Alzheimer's. Slowing aging pushes off multiple diseases at once, so the health and economic gains stack up much more significantly.
That's a powerful argument for prioritizing aging research.
It really is. And a more recent analysis, published in Nature Aging in 2021, put it in even starker terms. They estimated that slowing aging sufficiently to increase life expectancy by just 1 year would be valued at an incredible $38 trillion to U.S. society in present value terms, increasing it by 10 years. A mind-boggling $367 trillion.
Those numbers are almost unbelievable.
They are staggering. And again, they try to capture not just healthcare savings, but the broader economic contributions and the value of those extra healthy life years. While you can always debate the specific assumptions in these economic models, the fundamental message is overwhelming. The potential return on investment from effectively targeting aging is colossal.
What about potential downsides? Wouldn't people living longer put huge strains on things like Social Security and pensions?
That's a common concern and a valid point to consider. However, the proponents of the longevity dividend argue that these costs could likely be managed through sensible policy adjustments, perhaps gradually increasing the retirement age in line with increasing healthy longevity, for example. More importantly, they argue that the net economic effect would still be overwhelmingly positive. Because the savings from reduced healthcare costs associated with chronic diseases, combined with the increased economic productivity from people working longer and contributing more, would likely far outweigh the increased pension costs. It essentially improves the ratio of active workers to dependents, especially if older individuals remain healthy, independent, and engaged.
So it changes the whole societal equation.
Fundamentally, it could free up vast resources currently consumed by end-of-life care, allowing investment in other priorities like education or infrastructure. It enables older individuals to continue contributing their skills and experience for longer, making 60 the new 40 potentially a functional reality. It reframes aging from a period of inevitable decline and dependency to one of continued opportunity and contribution.
It really reframes the entire narrative around getting older.
It does. Of course, there are important ethical questions to address, which we'll delve into in later deep dives, things like equitable access to treatments, potential impacts on inequality, resource use, overpopulation concerns. But many economists argue that the gains in productivity and independence could alleviate some of those burdens.
So the conclusion from this perspective is that addressing aging itself is the most efficient and impactful path to combating chronic disease globally and improving overall quality of life.
That's the core argument of the longevity dividend concept. And given that the science shows aging is plastic in animals and we're seeing the first hints in humans, pursuing this goal seems not just scientifically exciting, but potentially one of the most beneficial things we could do for humanity.
Okay, before we wrap up this foundational episode, let's circle back to something crucial you mentioned earlier, the need for keeping it real. We need to balance the hope and excitement with a healthy dose of realism about the hype and the challenges ahead.
Absolutely essential. Our commitment throughout this entire series is to maintain evidence-based optimism. The potential of geroscience is genuinely enormous, but precisely because of that, the field has always attracted its share of hype, exaggerated claims, and unfortunately sometimes outright snake oil.
You see longevity clinics popping up selling unproven treatments.
Exactly. For every solid scientific advance, there seems to be a wave of breathless press coverage or companies making promises that vastly outstrip the actual data. So distinguishing the real, rigorous science from the wishful thinking or premature commercialization is paramount.
And that means being honest about the failures and limitations.
Critically honest. We have to embrace the negative data, the null results, the failed trials. They are just as informative, maybe even more informative sometimes, than the successes.
We've touched on some examples already.
Right. The failed Unity senolytic trial for osteoarthritis, a big setback for that specific approach in that context. The finding that metformin might interfere with exercise gains, highlighting unexpected complexities and trade-offs. The general failure of high-dose antioxidant supplements to prolong life in major trials, despite plausible mechanisms. The long string of failures for Alzheimer's drugs targeting pathways often linked to aging, like amyloid or tau, showing how hard neurodegeneration is to treat.
Learning from what doesn't work is key.
It's fundamental to scientific progress. Acknowledging failures prevents us from falling into confirmation bias, where we only pay attention to results that fit our preferred narrative. We have to be rigorous.
This leads perfectly into something you suggested we do throughout the series, asking the question, what would change my mind? It's a way to practice intellectual honesty and keep ourselves grounded.
Exactly. It forces us to define what evidence would actually shift our current beliefs or assumptions about the field. So let's apply it now to the overall prospect of modifying human aging.
Okay. What's your current working assumption based on everything we've discussed?
My current working assumption, which I think is shared by most scientists actively working in the field, is that aging is indeed biologically modifiable to a meaningful degree in humans. The evidence from model organisms, worms, flies, mice, even primates showing substantial extensions of lifespan and healthspan through various interventions is just too strong and consistent to ignore. It proves the principle that aging rate isn't absolutely fixed.
So you believe it's possible based on animal data. How confident are you about the magnitude and timeline in humans?
That's where the probabilities come in. If I had to put a number on it, my sort of subjective Bayesian prior, yeah, I'd say I have maybe a 70% confidence that interventions emerging from geroscience will add something like 5 healthy years to average human lifespan, health span within the next couple of decades. So a noticeable, meaningful improvement.
Okay, 70% chance of 5 healthy years soon. What about more dramatic effects?
I'm considerably less confident about radical extensions in the near term. Maybe only a 20-30% confidence that we'll see interventions capable of adding, say, 20 or more healthy years within our lifetimes. That feels like a much bigger leap, unless there's a truly revolutionary breakthrough, perhaps in something like safely controlling partial cellular reprogramming, which still feels quite far off.
So, reasonably confident about modest gains, less so about huge leaps soon. Now, the key question. What evidence would decrease your optimism? What would make you revise those probabilities downwards?
My optimism would take a significant hit if we see multiple large-scale, well-conducted human trials of different promising geroprotective interventions consistently fail over the next 5-10 years.
Consistent failure across different approaches.
Yes. For example, if the TAME trial shows no significant delay in the composite disease endpoint with metformin, and if upcoming phase 3 trials of rapamycin analogs show no meaningful improvement in immune function or reduction in infections, and if the next generation of senolytic trials also fail to show clear benefit in relevant geriatric conditions, if we see a pattern of failure across different mechanisms and different drug classes,
what would that imply?
It would strongly suggest either that human aging is fundamentally more complex and resistant to intervention than aging in model organisms, or that we've fundamentally misunderstood or mistargeted the key driving processes in humans. It might lend support to a more pessimistic view, perhaps the null hypothesis that aging is just an incredibly complex emergent property of entropy and accumulated damage that cannot be significantly slowed without causing massive unintended negative consequences due to things like antagonistic pleiotropy, where genes beneficial early in life become harmful later. If even multi-target interventions fail to move the needle in humans, that hard constraint view might gain more traction.
Okay, so consistent failure across trials would lower your confidence. Now, conversely, what evidence would increase your optimism? What would make you think, wow, maybe those radical extensions are possible sooner?
My optimism would skyrocket if even a single, well-designed human intervention trial yields unambiguous, clinically significant positive results across broad measures of aging or health span.
Just one clear win.
Yes, a clear proof of concept in humans would be transformative for the field's outlook. Imagine, for instance, a 5-year randomized controlled trial in adults aged, say, 70, 80, and the group receiving a specific intervention, maybe it's a rapalog, maybe a senolytic cocktail, maybe something else entirely, shows a 50% lower incidence of the composite outcome, cancer, heart disease, cognitive decline, death, compared to the placebo group. And maybe they also show significantly better physical function, like walking speed or grip strength.
A home run result.
Exactly. A result like that would provide definitive proof of principle that substantial geroprotection is achievable in humans. It would instantly shift my priors towards a much higher probability of significant health span extension being feasible. Even a clear success in a long-lived non-human primate, like demonstrating robust reversal of multiple aging hallmarks and lifespan extension using partial reprogramming in monkeys, would also significantly boost optimism about eventual human translation.
So the next decade or so, with results from trials like TAME and others coming in, will be absolutely critical for shaping our understanding of what's truly possible.
Absolutely critical. We're moving from an era of primarily animal evidence and mechanistic speculation into an era of empirical testing in humans. Science is a Bayesian process. We start with prior beliefs based on existing data, and we update those beliefs as new evidence comes in. We have to be prepared for surprises, both positive and negative. Aging research has certainly delivered plenty of surprises already. Well, the initial discovery in the early 90s that single gene mutations in tiny nematode worms could double their lifespan was revolutionary. It shattered the long-held dogma that aging rate was immutable. More recently, the realization of the profound impact of cellular senescence, those zombie cells, and the inflammatory environment they create was another major shift. The field is constantly evolving.
So let's try to bring all this together as we conclude this orientation. We've learned that aging isn't just one thing. It's this complex constellation of interconnected biological processes. The hallmarks, everything from DNA damage and epigenetic drift to failing protein quality control, mitochondrial dysfunction, stem cell exhaustion, and chronic inflammation.
Right, a multi-layered breakdown of maintenance and repair.
But the core message of geroscience is that these processes are, at least in principle, actionable. They can be targeted and potentially modulated to slow down the overall aging process.
That's the central premise, yes. And we now have increasingly sophisticated tools to measure biological aging, like the epigenetic clocks, which are vital for research and might eventually become validated surrogates for clinical trials, though we're not there yet.
We also have this rapidly expanding toolbox of potential interventions. It ranges from the foundational lifestyle factors like diet and exercise
That remain incredibly important.
to repurposed drugs like metformin and rapamycin that are already in human trials, and further out, next-generation therapies like senolytics, gene therapies, and even that futuristic partial reprogramming concept.
A whole spectrum of approaches at different stages of development and validation.
And the early human trials, while sometimes disappointing, are also showing these tantalizing glimmers that aging is modifiable in us. We've seen hints of improved immune function, slowed epigenetic aging rates, but they also underscore the complexity, the potential for trade-offs, the need for caution.
Absolutely. The potential reward, a healthier, longer-lived population enjoying that longevity dividend, is so immense that it absolutely justifies the rigorous scientific pursuit. But that same potential reward demands an exceptionally high standard of evidence. We have to avoid rushing ahead based on hype or incomplete data.
So throughout this series, we'll keep coming back to that dual focus,
Yeah.
presenting the best available evidence, citing the key studies, but also being very clear about the gaps, the unknowns, the null results, the ongoing debates.
Exactly. By the end of this series, our aim is for you, the listener, to have a really deep, grounded understanding of the biology of aging and an honest, evidence-based appraisal of how close we truly are to translating that understanding into practical, safe, and effective medicine for extending health span. This orientation today provides the essential concepts and, hopefully, that healthy dose of scientific skepticism you'll need for the deep dives ahead.
It really feels like the story of aging science is being written right now, chapter by chapter. And if we stay guided by rigorous evidence, keep an open mind, learn from both successes and failures, who knows, maybe we will write that chapter where aging itself becomes, in many ways, a treatable condition.
And perhaps where the vibrancy and capability of that 80-year-old ultramarathoner we started with becomes a much more common rather than exceptional reality for many, many more people in their later decades. That's the ultimate hope driving this field.
Thank you for joining us for this foundational orientation. In our very next deep dive, we're going to keep laying the groundwork by asking a really fundamental question. Why do we age in the first place? We'll explore the fascinating evolutionary theories of aging, which provide a different but complementary perspective to the what and how we focused on today. Then, after that, we'll start systematically plunging into each of the hallmarks of aging in detail. And just a reminder, we're making supplementary resources available for this deep dive. We've got a simple one-pager explaining surrogate versus clinical endpoints, and also a sort of living table summarizing the different types of interventions we discussed and their current level of evidence. You can find links to those in the show notes or on our website, along with citations for the key papers and sources mentioned today.
Great. And before you go, maybe a little homework if you're interested. We invite you to start your own personal observational phenotype ledger. Nothing complicated, just a simple log where you track a few basic health indicators for yourself.
What sort of things?
Maybe things like your resting grip strength. Can you easily open that stubborn jar? Or maybe get a simple hand dynamometer. Or time how long it takes you to walk a familiar distance briskly. If you use a fitness tracker, maybe note down your average resting heart rate or your typical sleep quality score each month. You could also track your waist circumference, maybe relative to your height. And if you happen to have recent fasting blood glucose data, include that. So simple, practical measures of function and basic health.
Exactly. The idea isn't to run a controlled experiment on yourself, obviously, but just to observe your own baseline and see if any changes you might make in your lifestyle, perhaps inspired by things you learn in this series, are reflected in these simple metrics over time. It's just good practice to be aware of your own health trends, and it connects these sometimes abstract scientific concepts back to your own lived experience.
A nice way to engage with the material personally. The quest for healthy longevity is truly underway, and it's undoubtedly one of the most exciting and potentially impactful scientific frontiers of our time. We really look forward to exploring it in depth with you.